So of course, a small amount of it is converted to CO2 and ammonia by urease in the intestine, as you've seen. So that particular enzyme called urease works or breaks down urea to produce ammonia again. And we've seen that ammonia is not just necessarily bad to the body, it also has its own functions. Okay, so there are about 6 amino acids that participate in this cycle, what we are calling the urea cycle. So ultimately, you're producing this particular molecule, urea, from ammonia. And the site of urea cycle is, or urea production is, the liver, and majorly mitochondria and the cytoplasm of the liver cells. So what are the steps? So in the first usually happens, remember, glutamine to the liver. Glutamine, you saw ammonia has been generated, it's converted into glutamine, so you should have that at the back of your mind. And once that has happened, then glutamine has gotten to the liver, and now ammonia is being released from glutamine in the liver. Once it's released, then it's ionized to become ammonium. So the first step, ammonium, CO2 are being condensed to form what we call carboxyl phosphate, and that particular process utilizes 2 ATP molecules, and you require an enzyme called carboxyl phosphate synthase 1. That is the enzyme that is being generated. So in the first step, carboxyl phosphate is being generated from ammonium and CO2. These two are being condensed, and ATP is utilized. Okay, that's the first step of the urea cycle. The second step, citrulline is being synthesized from carboxyl phosphate and ornithine. So ornithine and carboxyl phosphate are now combined with the help of an enzyme called ornithine carboxyl transferase to generate a molecule called citrulline. That's the second step. The third step, we are producing a molecule called adenosine, and adenosine is being generated as a result of the condensation between citrulline and aspartate. So when you combine citrulline and aspartate, you produce adenosine. It requires ATP and an enzyme called adenosine synthase that produces adenosine. Once you have adenosine, then it's cleaved, it's broken down into arginine and fumarate. It is broken down into arginine and fumarate, and that particular cleaving is done using adenosine layers. So adenosine layers cleaves adenosine so that we then produce arginine and fumarate. Then arginase cleaves arginine to produce urea and ornithine. Okay, arginase cleaves arginine. No, arginine is an amino acid to then produce urea and ornithine. Of course, we cannot be worried about fumarate. We know what will happen to fumarate. It's just an intermediate within the TCA cycle, so it can be utilized in energy production. But once we have arginine, then arginine will yield urea and ornithine. Okay, so this is exactly what is happening in the urea cycle. Picked this from the Harper's Chemistry by Harper. So what is happening here is that the first step, you remember ammonium being released from glutamine is combining with CO2, this case. We said this process requires about two ATP molecules. with an enzyme called carbonyl phosphate synthase 1. It is happening in the mitochondria. It is generating a molecule called carbonyl phosphate. That is what is being generated. And still in the mitochondria of the liver cells, ornithine is combined with carbonyl phosphate. Ornithine is combined with carbonyl phosphate to generate a molecule called citrulline. Citrulline. Okay. Then, of course, that requires an enzyme called ornithine transcarbamylase. All right. Then citrulline is combined. Now we move to citrulline moves out of the mitochondria and gets to the cytoplasm. So citrulline condensed with aspartate, okay, will produce adenosine. And this requires an enzyme called adenosine synthetase, and this again requires ATP. So once you have adenosine will be cleaved by adenosinease to produce arginine and fumarate. Okay. Then of course, arginine will again be cleaved using arginase to produce ornithine and urea. So you're having urea and ornithine. So this is the cycle. Ornithine, of course, will again be combined with carbonyl phosphate to produce citrulline, and that is how the cycle goes. Okay. So of course, we know the importance of the urea cycle. We've seen that accumulation of ammonia in the body is not good. We've looked at that process. The condition is referred to as hyperammonemia. And hyperammonemia is not a good, it's dangerous to the body because you see that it leads to death. It can ultimately lead to death. So urea cycle is important because then it's helping us eliminate ammonia from the body. But you also see that in the process, we also eliminating nitrogen that is excess in the body. But arginine is also being produced, and we know arginine is an amino acid. Okay, it's key in the protein synthesis. But once you break down arginine, of course, you break down arginine, you can produce urea and ornithine. Also, so that this can produce fumarate, and fumarate can take part in TCA, but can also undergo gluconeogenesis. So, urea cycle is also important in that sense. It can still generate energy. It can be utilized in generating energy. Much as it's an energy utilizing process, we've seen about 3 ATP molecules are being utilized. In the first step, 2 ATP molecules. I think in the third step, 1 ATP molecule is being generated. If I'm not wrong, yeah. If you look at this process, the combination of CO2 and ammonium to produce carbonyl phosphate, 2 ATP molecules are being generated here. And once you have carbonyl phosphate and ornithine, no ATP is utilized. Once you have citrulline combining with aspartate, ATP is being utilized here, 1 ATP molecule. So yeah, this is 1, these are 2, these are 3 molecules being generated. But then, since we have fumarate being generated at this particular point, we can then still utilize this in the Krebs cycle to produce energy. Alternatively, it can be utilized in the production of glucose via gluconeogenesis. Okay, so those are the key things that I wanted us to look at